All right, it's yours.
Yeah, so today I'm going to discuss some recent progresses in the field of oral macrocyclic peptides. So there are quite a few things happened in the past month or two, but today I'm going to highlight two events. One is Merck published their phase 3 trial of their PCSK9 inhibitor. This is the oral inhibitor Enlicitide, which is called CORALreef Lipids. The second event is Novartis just started a collaboration with Unnatural Products on their synthetic macrocycle platform. I'm sorry, I think there's some format change in here. Next slide, please. Yeah, the first CORALreef Lipids trial, this is a random control trial. So they use two different populations of patients. One is the people who already have cardiovascular diseases with LDL of 55 milligram per deciliter or higher. The second type of patients are people who don't have the CV diseases yet, but they're at high risk for developing this type of disease. And this group have a LDL level of 70 milligram per deciliter or higher. So on the right is the results. So the primary endpoint, so the patients take this pill once daily. The primary endpoint is the LDL reduction at week 24. As you can see, at week 24, Enlicitide reduced the LDL by 57%. The placebo group had about 3% increase. And the adverse event rates are pretty similar. So this is essentially monoclonal antibody type of efficacy using an oral agent. So this is really strong validation for the modality of peptides. So in the similar setting, PCSK9 monoclonal antibodies typically reduce 50 to 60% LDL in this setting. This is on top of the standard of care. So this is not mono agents. Nevertheless, the oral agents achieved the same level of efficacy as monoclonal antibodies. This is a really strong push for the field. Next. So another piece of news was Novartis has started this collaboration with Unnatural Products. This is a small biotech in California. So their platform is synthetic peptide for a permeable macrocyclic peptide generation. So the deal contained $100 million upfront and 1.7 billion potential milestone payments. So there's not a whole lot of information about this platform. So I picked some information from their website. It seems like they're making fully synthetic macrocyclic peptides or unnatural products, they claimed. Based on the fact these founders were students of Scott Loki, people guessed this is probably based on solid phase synthesis. So on the bottom, on the left is basically their workflow. So they say they can take hits from display technologies like mRNA or phage display or even DEL. So they'll get hits from other technologies. And then it goes through this typical SUR development cycles, but they use permeability, cell permeability, as a key metrics to optimize. So what they claim was they can generate peptide, non-natural product peptides at scale. So basically, they can make a lot of compounds very fast, so they can reduce the cycle time and get a large number of peptides through the cycle and then get the permeable peptides. So on the right is basically what they illustrate. They can start from a somewhat random point, a starting point in the chemical space, and they can use this as their engine to hop to the drug-like space very quickly. So I think essentially they just can make a lot of pipesets very fast. So this is, next slide, please. So this platform apparently it get recognized by several major players. In addition to Novartis, there are other notable players in here like Merck. So this is probably another strong validation of the modality, although from a different angle. So this is basically the big pharma buy into the story. Enlicitide story was already proven in clinic. So both events actually provided a pretty good validation for peptide as a modality. Next slide. So why there's so much love for peptides? So in terms of molecular size, peptides are between small molecule and biologics like monoclonal antibodies. So this is equivalent to cars in comparison to bicycles and airplanes. So apparently, cars are a very useful transportation vehicle. So by the same analogy, peptide could be a very useful modality in therapeutics. So some people believe peptides are a Goldilocks modality. So as Muhammad Ali used to describe himself as a float like a butterfly, sting like a bee. So peptides in terms of size, so they could flow like small molecules. They can pass through membranes, can be orally available. They can get to the disease tissues deep and fast, but they can still sting like monoclonal antibodies. They can still bind the target with antibody-like potency and selectivity. So this has been proven by Enlicitide story. And also, compared to small molecules, peptides are proteins' evolutionary partners. So nature uses a lot of protein as ligands. So this is another advantage of peptide. A practical aspect of peptides so the metabolites for peptides are typically pretty simple. It's because it's mostly just the hydrolyzed product. So they're typically harmless. So this is this can prevent drug-drug interactions that can be an issue for small molecules. And lastly, peptides are very easy to make. There's only one bond, amide bond, to be formed just repeatedly. So this is another big advantage. But there is some baggage for peptides. The first one is because peptides are connected by amide bonds, and there are plenty of protease in the system can hydrolyze that. So stability is really a problem. But now cyclization pretty much solved that problem pretty definitively. Another one, which is permeability, this is much harder to solve because the molecular size of peptides. Permeability can be a really challenging metrics to optimize. But there are some progresses in the field because of the advantages this modality has. So there are the different display technologies like mRNA or phage display, and also DEL is very mature technology now. So these technologies can generate really large peptide libraries. And there are some chemical advances like N-alkylation, incorporating non-canonical amino acids like the Unnatural Products platform. And macrocyclization is a really key. That technology really solved the stability problems and make really good progress in solving the permeability problem. Next slide, please. So why is macrocyclization so important? Mostly it's because the linear peptides have too many useless and harmful conformations. So the conformation is just a fancy word for molecular poses. So the issue at hand is for the targets that need peptide ligand. So these are typically undruggable for small molecules. So for these type of targets, if you want to be realistic, you need at least 10 mers, 10 amino acid peptide to get enough potency and selectivity. So the probability for a random linear 10-mer peptides are bigger. To have a measurable oral availability or cell permeability is technically zero because the hydrolysis by the proteases and the permeability. The reason is large molecule, the larger the molecular size, the more conformation you would have. Actually, the conformation increases exponentially. As shown in the boxes down below, this is just illustration. So say you have a large molecule, you have different populations of conformers or conformations. So the protein, the target typically just require a very specific conformation. Maybe you have one population that binds to the targets, that's the desired. But you might have another conformation that binds to the efflux pumps that will reduce the permeability and the oral availability. Another group of conformation will bind to the metabolic enzymes and get hydrolyzed or metabolized otherwise. And you have yet another population may bind to the off targets that give you side effects. But mostly, most conformations will be just unproductive. So it doesn't do anything. They don't do anything. They only dilute the effect concentration of the desired conformation population. And some of these conformation actually can shift because they're all in equilibrium. They can drain the useful conformation into unproductive pathways. So this is the fundamental issue facing the peptide field. Next slide, please. So macrocyclization is the most effective strategy to limit the number of conformations. So there are technologies available to actually do this. For example, in cyclosporine A case, the compound can form intramolecular hydrogen bonds that will reduce the number of conformation to make you pay less entropy penalty to get to the active conformation. But the most effective way is to form a chemical bond. In which case you would inject a lot of free energy into the system that will, once you form a bond, there's no way they can get back to other conformations. The larger the cycle, the macrocycle is, the more single bonds will be impacted. So these single bonds are where you get a rotation and create conformations. So that's why you see a lot of macrocycles. The larger these cycles are, the fewer the conformation you would have. And also some exopeptides recognize the terminals of peptides. So if you do head-to-tail cyclization, you will remove the terminals that can be recognized, and then that prevents the hydrolysis. So this is all good on paper, but when you have a lot of conformation, you want to keep just a few. It put a lot of pressure on design and screen because you don't know which ones are actually will give you the desired conformation. This is a typical finding the needle in a haystack problem. This is a problem in general for medicinal chemistry, but this for a large molecule like cyclic peptides, this is even a bigger problem. So next slide. So there are some really successful macrocyclic cyclic peptides. The most famous one is cyclosporine A. So this compound has 10 to 11 amino acids, but the oral bioavailability of this compound, it depends on the formulation you use. It could be anywhere between 20 to 60%, but people typically refer as 20 to 30% oral bioavailability. This is remarkable consider how big the molecule is. But this is really a gift by Mother Nature. The compound uses a so-called chemolionic mechanism, meaning before the compound binds to the target in solution in the system, it forms 4 hydrogen intramolecular hydrogen bonds. So that lock the molecule into certain conformations, and it also masks the polar surface area and expose the hydrophobic groups to make the compound very permeable. Once the compound binds the target, it opens the intramolecular hydrogen bonding interactions, and then the compound is in equilibrium between these two group of conformations. But this is extremely clever design by nature. Chemists cannot do this, at least routinely yet. There are a few other famous cyclic peptides like vancomycin and daptomycin. These are antibiotics. So vancomycin is also a very clever molecule. It functions as a receptor. It binds the diala in the bacterial cell wall tail. So typically, small molecules bind to the target. The small molecule is the ligand, so target is the receptor. But vancomycin is doing the reverse. Vancomycin is functioning as a receptor, and the bacterial cell wall actually is the ligand. And there's another one is somatostatins. These are well-known natural products. So when the Mother Nature is not helping, things can get really difficult. So this is the case for PCSK9 inhibitor Enlicitide. As I showed before, this compound achieved antibody-level efficacy, but it really took a lot of investment and effort to get to this point. As shown in the box, the screening hit was a 1 micromolar peptide with 14 amino acids. And then the Merck scientists took this monster and did a really tour de force MedChem optimization campaign to get to the final compounds. So this is actually a bicyclic peptide. The potency is 2 picomolar and is orally available. So this is unbelievable achievement as a medicinal chemistry program. So there are some other peptides are well known now. GLP-1 agonist like semaglutide is probably the most famous one, although this is not a cyclic peptide. And then L23 inhibitor, J&J's compound, is also close to the finish line. That is a cyclic peptide. And the Chugai has a KRAS inhibitor, Luna-18. So the field clearly is making progresses. But this is still a very difficult medicinal chemistry field. Next slide, I believe the last slide, please. Yeah, so going forward, so currently there are successes as shown in previous slides, but the field is still very challenging. Enlicitide, I don't think people should take this as a routine effort. You can get any program to this level of success. This is really a massive investment. Even with this level of expertise and investment, the compound still needs a permeation enhancer to help to get through the GI tract. Even with the enhancer, the bioavailability is only 1 to 2%. So it's still very poorly absorbed. So the problem for this is not only because the availability is low, it's also when the bioavailability is low, the individual variability can be high. So that could be a safety issue too. So now the field is learning, and Chugai just figure out some guidelines to make peptides orally available. So this is similar to Lipinski's rule of 5. So you don't want to have too many amino acids. You have to alkylate a lot of your amide nitrogens. The C-log need to be high, and you don't want any charged groups in the molecule. And the field also developed some enabling technologies like alkylation, macrocyclization, as we discussed, and the platforms that can routinely incorporate non-canonical amino acids. And there are platforms being built that can really produce fast SAR cycles and reduce the cycle time, like the UPN's platform. And the permeability enhancer field is also advancing pretty fast. So these things will increase the permeability of the GI tract temporarily when you give the drug. There's another older cyclic peptide platform called stapled peptides. So this technology will stabilize alpha helix so that they're useful for intracellular targets. The first stapled peptide went to clinic last year. So looking into the future, I think the peptide or cyclic peptides will be very useful modality in addressing non-druggable targets by small molecule or monoclonal antibody. For example, the protein-protein interactions, transcription factors, scaffolding proteins. So these are typically not accessible by monoclonal antibody, and they're not druggable by small molecules. So peptides will have a utility in these fields. And people now using peptides to make degraders. So hopefully that will expand the degrader field. And also peptides can be made in really at scale, and you can generate SAR very fast. So this is probably the most ideal subfield for AI in drug design. We can get a lot of SR initially and train the AI models. So hopefully we'll see AI will play a role in this field too. Yeah, so this is basically what I have. This is mostly chemistry oriented. If anyone has questions, I can answer.